else? We're way late, and I've got a couple that haven't shown up, so I got to get started. Let trying to think where that is.
Mr. Moore, so for the acid test, if I use glucose sugar in my lab, that could be a test chemical that I write down for the chart?
Oh, let me bring this up, and then we'll talk about it.
Okay.
Okay, ask your question again.
For the acid test, and I use glucose sugar in my lab report, could I use that as the test chemical for?
Sure, because that's what might work.
Okay.
In a methyl red test, you have to use glucose sugar. But see here on this McConkey, I had putting neutral red as a test chemical. Neutral red is not the test chemical. It shows you the results, but it doesn't enter into the test. And there was several other indicators like I can't remember other indicators, but methyl red or phenol red were indicators, and some people put down. I believe there's an indicator in ornithine here. I don't remember what it is. Something violet. Broncresol, maybe. Now the other thing is over here. We always have a lot of people messing up this side because they want to put selective or differential in all these boxes. Now that was the first thing that we did, and I had to really hound you about the difference in those two. But selective and differential is just the first three. The rest of the not selective or differential. Mostly of them are chemical tests, like the acid test is just a chemical test. You got a chemical, it's sugar. Catalase is an enzyme, oxidase is an enzyme. Don't make this hard. Don't try to make it something. Just let it what it is, because you got in the note up here what these are. And of course, motility didn't have anything to do with chemicals.
Well, Mr. Moore, I have a question about that one for acid test. Can you explain or give? So I was under the assumption, which I was wrong, that if it changed color, it meant differential. So is that not how it goes?
See, on differential, you've got to have a colony plate. You've got to have a gel plate to where you can grow more than one microbe on it. See, you can't grow two microbes in a broth because they would get mixed up and you couldn't tell which one was which. So you can't do a differential test on broth.
Okay.
I know that I have seen some people list the acid test as a differential test because it will tell the difference in certain microbes, but you've got to do two tests to tell the difference in two microbes, and on a culture plate, like a McConkey plate, you can put 3 or 4 microbes on there and tell the difference in all of them as they go, in one test. So it's not my idea of a differential test anyway.
Hey, Professor Moore, I had a question. I kind of missed it, a methyl red one. Were you talking about the test results? So I put for test chemicals, I had methyl red and glucose.
So as long as you got glucose in there, I'll growl when I look at methyl red, but
Okay, I mean, I take it off. I just didn't like.
you'll get credit for having glucose in there.
Okay, so glucose is what you were concerned about. Methyl red, you just want to know that's the name of the lab, right?
But yeah, that's the name of the lab. But
Okay, I missed what you were saying. I had these pop-up come up, so.
Okay, so can I redo mine and resubmit it before you grade it?
yes, trying to figure out how I guess I'll always try to grade the last one. I've got 2 or 3 people submitting that paper again. Evidently I drilled it enough the other day when I said that, because I made all these statements before. Because it was a whole bunch of us didn't get it. So obviously I wasn't effective. Okay, anything else on this one? Want to bring up this flow chart again. Hope you've done some work with the flow chart, because this is the last time that we're going to be able to talk about it before you actually have to do it. The flow chart will be assigned with our class, the first class after the exam, which would be next Tuesday. The general unknown, which uses the flow chart, will be assigned then. It will not be assigned during class. It's not an in-class activity. We will assign it in class, and then it will be due the next night, I believe. You've got 30 hours at least to do it. It doesn't take too long to do. If you know what you're doing, it takes hardly any time, probably an hour, and you do it. I want to go back over it just a little bit. Now, here was the flow chart for gram-negative organisms, and here was the way I listed this for you to look at. And remember that the order of the test that you look at, you don't look at this order that the way they're listed down here. That has nothing to do with it. The order comes from your flow chart. So right now, I'm going to show you the other way, the way that I'm actually going to give you these unknowns. I'm not going to give you this chart like this. Now, what I did, I'm going to try to leave that corner and this down here, and maybe you can see it. But what I have done on your unknowns, I've made a paragraph. But the paragraph has exact same information here. So I said an unknown gram stain that appeared as pink rods. You see here the gram stain was pink rods. Macaque plate had a pink growth. Okay, you go up here on the macaque and you see pink growth. The macaque plate had a pink growth, but the PEA did not. The PEA was no growth, no colonies. All right, you get what I'm saying? This paragraph says exactly the same thing as this chart. So I'm going to give you these unknowns. I told you there's going to be 8 of them. There'll be 8 paragraphs. Now, don't look at the paragraph and say, oh, I'm supposed to do this next. According to this paragraph, they have the gram stain, and now macaque, and now PEA, and now oxidase, and now catalase. Oh, you look at the flow chart to tell you what is next. Some of these things I write in here, there's be some distractions here that don't have anything to do with this test. So you're going to say, well, how do I use this one? You don't. It's not even involved. I mean, like on these gram negatives, you don't even use a catalase test. You don't even use an oxidase test. Well, I take it back. You could use an oxidase over here. But you never use a catalase test in this flowchart. So just because it's in the paragraph does not mean you're going to use it. What tells you whether or not you use it is following the flowchart. All right, let's just say that we had gram negative rods. Okay, going this way. And let's say that it says your McConkey test had growth. Okay, that's good. But it had no color, or it didn't say anything about color. So you'd know that this was lactose negative. So you'll go over here. Well, it just so happens this is a coincidence that the next step both ways is the methyl red. But so you do have the red, you need to look at the methyl red. If you say the methyl red is positive, your next step is decarboxylase. But if the methyl red was negative, next step is oxidase. So you let the chart tell you what you're looking for in this paragraph. Is everybody clear with that now? You do not look at the order. You do not even care what's in this paragraph unless it tells you to do that test. So if this test said gram negative rods and it said you had a pink growth on McConkey, well, that means you're positive, right? If your methyl red was negative, that means you come this way and your next step is orthidine decarboxylase. I can't say that word tonight. But if your methyl red was positive, your test is zero. Something happened to my chart here. It got messed up. Okay, so you get the idea. All I wanted to do was make sure you understood that I'm going to give you these in a paragraph form. It's no different than seeing it in a chart form, but I wanted to show you this way the first time I looked at it, and after we're sort of comfortable, then I wanted to show you the paragraph form. So what we'll have is, of course, tonight we review, and we'll have an exam on Thursday. Next Tuesday, we'll have a chance to make sure we know how to do this again, and the assignment will come on board probably about the time we leave out of class. It'll probably come on at 8:30 or something like that on Blackboard. And then you'll have then and the next day and maybe the next to work on it. I'll give you time. And you'll have time, if you're working, you'll have time to work. There'll be plenty of time. Anyone want to look at the flow chart again? Maybe look at the gram positive flow chart. There's some different things in the gram positive based on the gram negative.
Professor Moore, I just wanted to know with the unknown when we do that, once we do the flow chart or we figure out whatever it is, would you want us to write the report how it's written down at the bottom as well?
Right here. This is the way your report is written.
Okay.
And you have two instances of this. And what is always missed is the confirmation test. The confirmation test, remember, it's always listed down here underneath the names. The confirmation test is the last thing you say. The step right before, for instance, look at this one. The ornithine carboxylase, negative or positive, tells you what the organism is. If it's negative, it's Proteus vulgaris. Okay, the confirmation test is positive for endo. Remember, that would be the red ring. If here your ornithine decarboxylase is positive, then you're saying it's Proteus mirabilis, and endo negative is the confirmation test. So that would be the last thing that you state. How in this one, endo is the last test. That's several times you see that the second test is positive and negative on your confirmation. I don't know if y'all are seeing this, but mines keep changing on me. It's something to do with my screen, I guess. They're back now, so it's okay. Anything else you want to check out on the flow chart? The flow chart is simple. Let it work for you. Okay? Let's go into the review. What I'm thinking of it, we have a lot of people that use Blackboard. Let me see nods or waving or something. Okay, a lot of people use Blackboard. Okay, I will have an answer sheet on Blackboard. It may not be exactly perfect. I'm not going to have a lot of time. I have an answer sheet like we used before that I think had 36 answers on it, and then like 8 discussions. It will be similar to that. If I happen to give you a 36 answer sheet and we have 32 questions, don't worry about it. Just fill out 32 and go on. And if the numbers don't match up on the discussion, change the numbers and make it work. I have to take my son to the airport tomorrow. He's been down here for this holiday weekend that we had with the birthday party, and he has to go back to Austin, and I don't know how much time it's going to take me to do that. Otherwise, I have plenty of time to get all this set up for tomorrow, because my Wednesdays are almost always reserved to set up my exams. Okay, lecture exam 3. A lot of it about genetics in this particular test cycle. When we talk about genome, you look down here and the chromosomes they make up the genome. So the genome is all the genes of any organism. Could be ours, could be a plant, could be a bacterium. Now, when we talk about genome or genes, there are 2 terms we use. Of course, DNA we use all the time now. The other term is chromatin. Chromatin was the name for DNA before DNA existed. Chromatin is nuclear material that is genetic. Euchromatin is considered to be true genetic material, true chromatin. It's loose, is accessible, it's able to be used, and it's really most of our genes. Now, euchromatin and heterochromatin is only in eukaryotes. Hey, I'm lying to you. Euchromatin is loose susceptible DNA to be in eukaryotes or prokaryotes because it's just DNA. Heterochromatin, which is very tightly packed and wound up, like you see some of these components wrapped around proteins. Heterochromatin is only found in eukaryotes, but it's the tightly wrapped portion of gene in eukaryote. Eukaryotes also have euchromatin, so it would be the chromatin that's part of the gene. So euchromatin is true chromatin no matter what kind of organism it is in. Heterochromatin is tightly packed. Much of it is not available for use in eukaryotes. Gene expression simply means to activate a gene. If you activate a gene or you turn on a gene, what you're doing is initiating transcription. See, that's the first step of gene expression is transcription. Transcription, of course, is where you make a copy of the original DNA. Now, the best way to say that is for what transcription is DNA code is converted to RNA code. Now, what's going to result is messenger RNA. So transcription is going to make messenger RNA. No matter what else you say about it in your descriptions, transcription gives you messenger RNA. Translation takes that messenger RNA code and changes that coding to a sequence of amino acids. And it gives you the order of those amino acids. So see, it's not just the amino acids are in there, but it gives you the order that they're arranged in. Because if you change the order of the amino acids, you're changing your protein.
More.
Yes, go ahead.
Sorry, I'm missing one. I got takes messenger RNA, converts it to what?
An amino acid sequence, or now think about this, an amino acid sequence is a peptide or it's a protein. When you say it converts it to an amino acid sequence, there are several things that amino acid sequence can to you. The simplest thing is peptide. But you get a bunch of peptides together or a long peptide and it may be a protein. This is a lot of times our handicap in this course is because no longer have we had to have biology, and so we don't know this terminology. Okay, so gene expression is transcription and then translation. Transcription is making messenger RNA. Translation is using messenger RNA to make an amino acid sequence. Now, when we're making an amino acid sequence, we're going to copy the DNA code. The DNA code on one side is one thing, complementary on the other side. So it must be designated which strand of DNA is going to be copied. Because see, this copy is going to be different than if it copied this side. It's going to be complementary to it. So the side that is being copied is called the template. So DNA template is simply the strand of DNA that is being copied to make messenger RNA. Okay, does everybody see that copying this side is going to give you a sequence if you copy this side? Everybody's okay with that? Because these are not the same copies. These are complementary to each other. So these two strands of DNA are different. True, they're just opposite of each other. But if I'm going to make a copy of this one, it's going to say AUC. If I make a copy of this one, it's going to say UAG. So the template strand is simply the strand of DNA that your polymerase copies to make your RNA. The triplet code is any 3 nucleotide sequence. It's 3 nucleotides in a row. Guys, every 3 letters that you can make out of A, T, C, and G is in this sequence down. This is all of the possibilities of 3-letter code A, T, C, G. Now, U is substituted here because it's messenger RNA. We should say A, U, C, and G. This is all of the codes that you can make. So these are all triplets, and every one of them has a meaning. So what is a triplet code? It's 3 nucleotides in a sequence that has a meaning. That meaning is, it stands for a certain amino acid. Now let's just go down to 10. The codon is a triplet code, that meaning. But it's always on messenger RNA. So a triplet code on messenger RNA is called a codon. And even more, a codon always stands for a certain amino acid. So no longer say amino acid, it stands for a certain, a specific amino acid. So AUU is a codon. AUU always stands for leucine. CGA always stands for arginine. Now, I think there's some others that stand for arginine, but CGA always does. So that's when we say a codon, you're getting down to specifics. If I say triplet code, you're not necessarily at a specific. It's just any 3-letter nucleotide sequence that has a meaning. Complementary code, that's just the matching sequence that you have. So complementary just means that it's going to be the nucleotide that matches the opposing nucleotide. So that means A always matches with T, C always matches with G. Those are complementary. Number 7, RNA polymerase. That's exemplified here in the picture by a big old sort of a triangular looking blob here. RNA polymerase is a huge enzyme that creates complementary codes. But the complementary code is RNA. So see, the RNA polymerase takes the coding from DNA and creates a complementary RNA code. In other words, it's making messenger RNA. And number 6, you just have pairings. A binds with T in DNA, A binds with U in RNA. G always binds with C, no matter what kind it is. So I'm sorry, I skipped it. So 6 and 7, RNA polymerase. Number 8, an RNA transcript. That's just the copy. See this red sequence here? That's a transcript. It's the sequence that's made from transcription. So transcription makes RNA. So there is an RNA transcript. It's just a way that we talk about what is produced by the messenger, by the RNA polymerase. And number 9, messenger RNA, if this were a bacterial and we're doing a copy of this DNA, this first transcript would be messenger RNA. Because in prokaryotes, in bacteria, the first copy of material is messenger RNA. Number 9, in prokaryotes, the first copy is messenger RNA, a primary transcript, the first copy. Now, number 11, in eukaryotes, the first copy here is not going to be messenger RNA. The first copy is going to be pre-messenger RNA. So what does that mean? What is pre-messenger RNA? The first copy has way too much stuff in it. The DNA does too. See, the DNA has extra things in it in eukaryotes. We said they had heterochromatin. Okay, so the extra stuff to be taken out. The regular good stuff is in number 12, the exon. This is the true coding that we need. So the exon is the sequence of its DNA and the sequence of RNA that's produced that has to be kept. But the number 13, the intron, is the sequence of messenger RNA that's extra. And so we look at this graphic that is on the screen. This pull up here is DNA. See, the DNA has the exons and introns. And so the first copy of transcript, the primary transcript, has the complementary copy of good and the bad. So this is pre-messenger RNA. It has to be converted. This is called DNA processing. Put it in great big letters with stars by it, or put it in a box, some way that you'll pay attention to it. The process that we're going to undergo to go from here to here is called RNA processing. We're going to process this pre-messenger RNA and it will become messenger RNA. So what is required to do RNA processing? We need a spliceosome. That's number 14. What is a spliceosome? It's a great big old monster thing that's made of proteins and RNA and enzymes, and it's just a great big dude that's going to take out all of the introns and hook up all of the exons to make messenger RNA. That sounds like a really good discussion question. What does a spliceosome do? A spliceosome causes RNA processing. It takes out introns from pre-messenger RNA and hooks up all of the exons to make messenger RNA. So here's a spliceosome doing its work. Once the messenger RNA is finished being produced, we put a nucleotide on the front of it. Notice this G here. It's just one nucleotide on the front to protect it. It's a guanine, but it's sort of different because it's got 3 phosphates. It's really GT is what it amounts to. But here it is on the front. It's called a 5' cap because the front of it is a 5' end. Just like we were talking about 5' directions in the test before this, it's still everything goes in a 5' direction. Okay, so that's the 5' cap. The other end of it just shows 2 of them, but there's about 250 of these put on the end. It's the poly A tail, just a whole bunch of adenines or adenosines. There's a whole bunch of them stuck on the end of it. It seems like that the tail has a greater chance of degrading than the head for some reason. Otherwise, they wouldn't have put so many nucleotides on the end of it. Okay, that's the end of transcription. That might have been something that I should have had in our review. You know, a little box around where transcription starts and where transcription ends. Because from now on, it's going to be translation. Transcription occurs in the nucleus, translation occurs at the ribosome. So the ribosome is the next thing in order here. The ribosome is an organelle where translation occurs. That's where protein synthesis occurs. That's where you make proteins. So this is a recap of what we've just done. So here is our DNA, and which strand would this one be right here? Somebody answer this. What is the name for the strand DNA?
The lagging strand.
No,
Oh,
leading and lagging were on the other cycle.
Is that the DNA template?
Exactly. There's your template DNA right there. That's the one that we're going to copy. So there is your messenger RNA, and actually it doesn't tell us here, but here notice it has a note about eukaryotes. And so you really can't tell with this one up here if it's DNA, it's messenger RNA or pre-messenger, but you can this one down here. Notice you have dark red and light red. So this is pre-messenger RNA. And here is where the splice is. We're going to take out these introns. And then here is your completed all red messenger RNA now. It was messenger RNA, but now it's messenger RNA. Now, this is nucleus. This is where transcription occurs. As we go toward translation, translation is going to be down here at the ribosome. Ribosome is a two-part little thing that clamps around the messenger RNA, and then the sequence on the messenger RNA is read by the ribosome. Now, I want to go to this picture for a moment because this is a version of what I was showing you a while ago. But what you see out here are a bunch of the transfer RNAs. Remember, you have to match up with the correct amino acid. And these transfer RNAs are specific for each amino acid. On the bottom of these transfer RNAs is the opposite code that we call that complementary to the what's on the messenger RNA. So let's get a picture of that. Okay, so we see a code here, UGG. That's a codon. Okay, UGG is a codon. If we wanted to know what amino acid it would pick, we would have to find it in this chart. UGG stands for tryptophan. Hey, that's one of them that we were talking about, in our lab. Okay, so this transfer RNA had a tryptophan on it. It's right here because it was hooked right here, and it's the one right before here. So this one was carrying this one. This one's carrying that. This is carrying here. So what would this amino acid be? We look at UUU, go back to the chart. UUU is where?
At the top.
Top left corner.
Ah, there we go. Phenylalanine. NutraSweet, y'all. That's what phenylalanine is. So you could just keep doing that and you could figure out what each one of these amino acids were. And if you had a lot of charts that the research geneticists have now, you could take the sequence of amino acids and figure out what protein it was. Because all of that stuff is on record now to know what your body is made of and what each gene codes for. So see all of this stuff that we're looking at, this is just the beginning of what geneticists know in order to figure out the genetic code, what each particular gene codes for throughout your whole body. And they now have catalogs, not for this, just for people, but they have it for animals, snakes, trees, bacteria, viruses, everything else. They have spent about 40 years now figuring the code of all these different organisms. And so you go and have a stool sample. They want to find out what's causing your diarrhea. A stool sample, do a genetic analysis on it, and find out if it's a virus is causing it, if it's a bacteria causing it. Because they have a record now, a catalog of each of your genetic sequences of microorganisms as well as us. So see, that's the utilization of these principles. Now, what's this dude called right here? This is number 18, the transfer RNA. See if I've got a bigger picture of it. I guess maybe this one's the best. Okay. These are transfer RNAs. Transfer RNAs have amino acids on one end and a code on the other end. And notice this code is complementary to the codon. So that's the way you call this. You call it an anti-codon because it's complementary to your codon. So transfer RNAs have anticodons on one end that matches them up to the messenger RNA. They carry an amino acid on the other end. So all of these transfer RNAs are specific, just like all of these codes are specific. Epigenetics is the study of environmental factors on gene expression. They studied these by studying identical twins that had been separated, and they began to see there were some differences in their genetics based upon where they were raised, kind of nutrition they had, and so forth. Sometimes it's disease, deficiencies, sometimes obesity. All of these things can have a factor or effect on gene expression. Proto-oncogenes are genes a normal cell cycle. A normal cell cycle has a G1 phase, a synthesis phase where you make replication of your DNA, a G2 phase that continues to grow, and then you have mitosis. Okay, this is the normal process of cells in your body. So they have a lifespan and then they divide. Some cells divide quite often, like your skin cells, like your hair cells. Some cells don't ever divide, like your muscle cells and your brain cells. If you have an abnormality that causes something like a muscle cell or a brain cell to begin dividing, that creates a tumor. So a proto-oncogene is a gene for a normal cell cycle regulation. If everything is working good, your proto-oncogenes are working well. They're causing the cell cycle to behave as it should. If a oncogene is mutated, it may become an oncogene which no longer can control the cycle like it should. So now the cell cycle might go out of control because it does not have the normal restraints on it. An oncogene has the potential to increase your cancer risk. Notice that the definition does not say that it causes cancer. It increases the risk of cancer developing. Another very specific kind of gene, besides the proto-oncogene, is the tumor suppressor gene that is a protector of the genetic code. It can protect or repair issues that occur. So tumor suppressor genes reduce cancer risk. You might say that tumor suppressor genes are anti-oncogenes. See, they would prevent oncogenes from causing you to have cancer. There are two types of tumor suppressor genes that I want you to know. One is called p53. It's very common in organisms. We have one. I believe we just have one. The other is the BRCA. It's a breast cancer gene. We have two. BRCA1, BRCA2 is what they're called. They're on different chromosomes even. BRCA genes are tumor suppressor genes, just like p53 are tumor suppressor genes. When are tumor suppressor genes not good is if they're mutated. Mutated tumor suppressor genes, okay, they take away your protection. So if you have a mutation that creates a cancer cell, there is nothing to protect against that cancer cell becoming a tumor itself. So mutated tumor suppressor genes increase your risk. Well, they increase your risk because you have less protection. But nowhere does it say that they give you cancer. If you have a firehouse down the street from your house, your chances of your house being destroyed in a fire are much less, right? If the firehouse closes, your risk of losing your house to a fire is greater, but you may never have a fire. Think of that as a tumor suppressor gene. Tumor suppressor genes increase the chance that if you have a cancer cell, those cancer cells will be destroyed. You may never have a cancer cell, and your tumor suppressor gene being mutated may never cause you a problem. The difficulty is most of us have cancer cells frequently. and our processes usually take care of them. Transposons are just whenever a gene sequence gets moved in your genome in some way. There are some processes called crossing over, and you don't have to write down, but several processes where genes can move from one place to another. If they do, they're called transposons. It just means they moved. Number 24, phase transduction. This is the same thing. I've been harping on it all semester long. Phase transduction. Now, remember that transduction, we have this vocabulary word later on. Transduction is the viral infection that puts genes in a new host. And phage means we're talking about bacterial. So number 24 is virus infecting bacteria with some kind of lysogenic infection that's going to go into their genome. Nosocomial infections are hospital induced. And think about hospital meaning any kind of institution or facility that is used for medical intervention. So nursing homes, dentist office, clinics, as well as hospitals. Reservoir of infection is just a natural area where microbes or not microbes, where pathogens reside. A natural area where pathogens exist. It could be an animal like a bat. It could be water. It could be soil. It could be food. It could be a person. A portal of entry is simply the method by which pathogen enters your body. It could be like a cut or a wound. It could be a receptor match, like the way coronavirus enters our AIM2 receptor. You guys that have had AP2, we talked about this. There is a receptor in your vessels, your capillaries, that coronavirus binds to. If we look at methods of disease transmission, contact is probably the most efficient way that microbes move from place to place. Parent-child, siblings, fomite is the way of inanimate objects. In the medical facility, this might be dressings, bandages, bedding. It might be tools, catheters, instruments. Droplet is pretty self-explanatory, coughing and sneezing, but also through talking. Kind of vehicle, which makes a method of transfer, such as a vector, like insects or ticks. Zoonotic transfers, and this is usually by rats and mice. Maybe carriers, which are unsymptomatic people. Asymptomatic. There's no such word as non-symptomatic. Important terms: selective toxicity. The ability of some kind of medication or some kind of treatment to kill pathogens without harming most. Selective toxicity. Virulence factors, the ability to produce pathogenicity. That's what virulence is. Several factors that we list here, and there's probably this many more. Flagella can be a virulence factor. Those remember the hair-like appendages for locomotion. Fimbria, which are little spiky hair-like extensions for attachment. Capsules, which are coverings around microbes. Enzymes. Enzymes can make us sick. Enzymes can do a lot of different damage to bodies. Toxins. We have endotoxins as well as exotoxins. Remember, endotoxins are gram-negative outer membranes, LPS and so forth. Exotoxins are actually secretions. And then phage infections. Phage infections will cause microbes to have different kinds of virulence. Like there was a Corynebacterium that was normal until it was infected by a virus. The virus gave it genes for the diphtheria toxoid. So it gave a gene to this Corynebacterium that was harmless. Now it is harmful because it creates diphtheria in its hosts. So see, phage infection can change microbes from being non-pathogenic into being pathogenic.
Professor,
Yes.
just want to make sure. So for everything to 30, we just need to know as examples of virulence factors.
Mm-hmm.
Okay.
We talked about a whole lot more detail before. When we talk about diseases, there's several different stages. An acute disease is one that you get sick and get over it, so your immune system is able to overcome the disease. It may make you sick. It may not make you really sick. But either way, the acute stage is illness, and then you recover. A chronic disease will have persistent and maybe intermittent times of infection. So it's persistent, it's chronic, something you have over and over or for a long period of time. Lysogenic, of course, is disease caused from viral infections into your genome.
Hey, Professor Moore, I had a question.
Oh,
For one of these, you had I had put down remember the term, and then I don't have anything for that. Was there something that we needed to remember for that?
for lysogenic,
Yeah, I highlight things when I don't have them. It says remember the term blank.
I bet it was transduction.
Okay.
Does anyone have that?
I have prophase and provirus.
That's what I have too.
Well, okay. A lysogenic disease has a stage that's called provirus, and that's where the virus, the viral gene is in the host gene. Yeah, George, that's probably what it was. It was being sure to remember that when you have a lipogenic infection, it's called that you have a provirus. If it's a bacterium, you have a prophage.
Thank you.
And see, that would be a condition or a part of transmission. We'll get transduction in a moment.
Right, before we move on for other virulence factors, I don't have anything for enzymes, not an example.
We had things like streptokinase and coagulase, beta-lactamase, collagenase. They're all enzymes that can cause our body damage. Concept A, steps in transcription. All right, this is just a summary of what we were going over back in the vocabulary a while ago. So under transcription, remember that it happens at the nucleus or nucleoid. So it has to happen where the DNA is. First, you must recognize the template DNA. So you have template DNA. The RNA polymerase binds the promoter. The RNA polymerase creates a complementary copy. And then step 4, it adds the 5' cap and the poly A tail as protectors. So that's sort of a quick version of what happens during transcription. What happens in translation? First of all, translation must occur at the ribosome. The messenger RNA is bound by the ribosome. The transfer RNAs have to hook up to their amino acid and they get matched to the messenger RNA codons. And so the messenger RNA code is converted to an amino acid sequence or peptide sequence. And that's what's occurring. See right here. See the ribosome jumps over the messenger RNA. The codon calls for transfer RNAs that match. And they bring in the specific amino acid. Little sequence, I forgot that they actually have the correct amino acid listed. So that's a little better sequence than the other one I used a while ago. We wouldn't have had to go back and look them up. Exotoxins. This one's going to be on your exam. I remember it. Exotoxins are gram-positive secretions. AB toxins have two components, an A component and a B component. So AB toxins. The A is an enzyme That's the thing that does damage to you. So A is the enzyme component. B is the binding component. That's going to be the receptor match. So AB toxins are two-part. There's a lot of AB toxins: diphtheria, cholera, botulism, tetanus. These are all considered to be AB toxins. Cytolacin, that word means cell splitting. Cytolysis, cytolysine. Lysis is splitting. Cyto means cell. These are pore formers. Cytolacins are pore formers. The most common kind are hemolysins. Hemolysins break down blood cells, so they make holes in blood cells. Remember alpha, beta, and gamma. Alpha hemolysis makes a green. Did anybody look up to see if those are shown on the final review? I know they're there. I just don't know if the links still work. Or you can look up your own link. Alpha is greenish, beta is clear, gamma didn't do anything. Here are two really good hemolysis looks right here.
Hey, Professor Moore.
Yep.
What'd you say for cytolysis? Pore former?
Cytolysis, see, cytolysine, cell splitting, that's what that means. But what occurs is these microorganisms make a hole in the cell, and it allows water to go in, and it splits the cell. Oh yes, pore formers. This is what they actually do. Is they make pores in cells, make little holes.
Okay, thank you.
So even though cytolysine does not mean pore former, that's what these are. That's the effect of them. Cytolysis just means cell splitting, but it's water that splits cells. PBL is a different kind. Staph aureus uses to make pores in cells. PBL is just a different kind of pore former. Superantigens are not pore formers. Now, remember that's a separate place. See, AB toxins and cytolysins and superantigens are all out even with each other. So 3 different kinds of exotoxins. AB toxins have 2 parts. Cytolysins are pore formers. Superantigens are highly inflammatory antigens. They cause your immune system to have a super response. That's where the word super antigen comes from, because the super antigen is just a normal antigen. What's super is the response. This is what causes toxic shock. It can be Staph aureus, it can be strep pyogenes, but they have antigenic responses that your immune system has a tremendous response to. Cox postulates. Remember, this is just a protocol to connect a certain pathogen to a specific disease. You have to have the same pathogen in every case of the disease.
Professor, can you repeat that definition one more time, please?
I don't remember what I said, but in trying to connect a pathogen to a disease, you have to have the same pathogen in every case of disease. I think what I said, you're trying to connect a certain pathogen to a specific disease. You got something? Got it?
Yeah, thank you.
Okay, now let's look at the steps. Okay, so you have a diseased organism. You must isolate the pathogen from that diseased organism, and you must culture it on a medium, grow it on a culture plate. So you have to isolate it from a diseased organism and grow that pathogen on a culture plate. Now you take the pathogen from that culture plate, you inoculate a second host. Second host must get the same disease. And you must isolate the pathogen from the second host and grow on a culture plate. If you've got the same pathogen, the second culture plate, then you can say, yes, this pathogen caused that disease. So in other words, you go through 2 cycles of disease transmission. That's what Koch's postulates developed, a protocol to do that. It's just a process. Now, the disadvantage of that process is the fact that you have to grow that microbe on a culture plate, because we found that there are certain microbes that will not grow on a culture plate. And so that's the disadvantage. If you have an obligate intracellular parasite, remember that term? If you have an obligate intracellular parasite, they won't grow on a culture plate. So that's a disadvantage of Koch's postulates. Makeup of a prokaryotic gene. If you have a gene, there are upstream elements and downstream elements from the gene itself. So you look at the makeup in concept D, the gene sequence is right there sort of in the middle. And before it, you have promoter and operator. After it, you have terminator. So a gene is more than just the gene. Okay, so what we have here is a set of nucleotides that is the promoter. That's the place where the polymerase can connect. It hooks up to the polymerase. The operator would be a gene sequence that either allows the polymerase to work or it stops the polymerase from doing anything. So it's the control element. That's what the operator would be. It would be the control. It either induces, makes it go, or represses, makes it stop. If you do have transcription through that gene sequence, then you're going to have another set of nucleotides that is called the terminator. In other words, it would stop the process when it is complete. So these are the elements of a prokaryotic gene. Remember, this is the simpler form of genes. If it were eukaryotic gene, there's going to be several pieces of paper taped together to put all the control elements together because they just have so much more control. Now, all I really want you to remember about the E. coli gene is the fact that in this particular situation, if you have control elements like the operator, they will not allow production of something like an enzyme if there's no food for it to digest. So it's just, see, if lactose is absent, it's going to create a repressor that stops the whole process. There's no sense in making lactase enzyme if there's no lactose for it to digest. In the second, if lactose is there, it allows the operation to go to completion. You make your enzyme and you digest the lactose sugar, the microbe gets energy, and everybody is happy. All I want you to understand is there is a rationale, there is a reason why there are these control elements like operators. We have in our body too. They're just so much more complicated that you can't go like a sheet of paper like this. And also, you have to have an instructor that can understand it because it's way above my pay grade. Microbe defenses, the way microbes protect themselves from us. Now, this is more common sense than anything else. They break down our enzymes, or they break down our drug, our medications. So they'll use enzymes to degrade our medications. Beta-lactamase is one. See, beta-lactamase would break down penicillin. So you give this microbe penicillin, that breaks down the penicillin and sits there in the zone. Okay, so enzyme degradation. It uses enzymes to break down our drugs. Cell wall impermeability. They will throw up protection around themselves to prevent our drugs from getting into their cell. Now, first of all, gram positives are more impermeable just because they have a thicker cell wall. But you take a gram positive and put a capsule around it, it can be quite difficult to get medication in there. So they're just throwing up barrier. Alteration of target sites. We make a medication that hits a certain part of their body, they change their body. It's through evolutionary change, but the body changes so that the medication we're using doesn't work anymore. This happens a lot with their ribosomes. The fourth thing that they can do quite easy is to pump our medications out of their body. Little pumps, like same kind of pumps that we have. We have proton pumps and sodium potassium pumps. They'll have pumps that work against some of our medications. I wish I had an example, I don't. Okay, 4 ways that microbes protect themselves from us. Okay, we've got 6 ways that we try to attack them. Well, the first one is penicillin, because penicillin would break down their cell wall. It was a beta-lactam antibiotic that would break down their cell wall. So we try to penetrate the cell wall with things like penicillin, beta-lactam antibiotics. We try to destroy their outer membrane if they're gram-negative. How do we do that? We can do it with soap, with alcohol, with some other chemicals too. So we try to break their cell wall, we try to mess up their outer membrane, or we give them some things that would be like nutrients for them, but we give them messed up nutrients that won't work. That's called an analog. So instead of supplying them the correct molecule, we supply them a defective molecule. That's the analog. Let them build their components, and those components don't work. They're dead. They don't. Their cell that they produce doesn't function. The problem is we give them analogs of nucleotides. Another one, skip them down to number 5, we give them analogs of folic acid. They're called sulfa drugs. In either case, we're giving them a molecule that when they make their material, it's no good. And so the cell doesn't live. We very often try to interfere with protein synthesis. At the ribosome, you had your messenger RNA that made proteins, made amino acid sequences. If we can mess up their ribosomes, we can mess up their protein synthesis and they die. And the reason we can mess with their ribosomes so easily is because their ribosomes are 70S and ours are 80S. So we give them something that messes theirs up and it doesn't harm us. We can have selective toxicity because they have different types of ribosomes than us. The last thing is specific for fungi. In our plasma membrane, we have phospholipids, but also have cholesterol. In fungal cell membranes, they have phospholipids, but they have ergosterols instead of cholesterol. So we can use chemicals that attack ergosterols. It will hurt them and not us. The reason we do this is to try to create selective toxicity, because fungal cells are very similar to our cells. So it's hard to create a chemical, a medication that will kill them without harming us, because we're both eukaryotes. But we do have a little bit of difference in our cell membranes, and therefore we try to attack them at that difference in the cell membrane. Concept G, when it comes to viral growth, we don't have anything that works. So all of that stuff I wrote down there is basically useless. We really do not have anything that works. We treat the symptoms of viral infections and let our body destroy the viruses.
Mr. Moore?
Yes.
Just want to be clear. So as far as for our tests, it's for this question, we will put nothing works or
And I won't ask you to do anything because there's nothing, it's a wasted question. I'm not going to waste our time.
okay.
I mean, you do need to know that nothing really works, but there'll not be a discussion question on something like this.
Okay, thank you.
Okay, inside and on us, actually on the outside of us and on the inside of us, microbes grow. They grow in our mouth. They grow in the vagina of women. And in all these places, we have antagonistic interactions that go to where the good guys are in constant battle with the bad guys. But the place where this occurs most is in our gut, in this large intestine, a little bit in the small intestine, but most commonly in the colon. So that's what I'm going to deal with mostly on this concept. Indigenous microflora just means microorganisms that are normal to our gut. Organisms that normally grow in the human gut. First of all, with infants, they have to be inoculated. It's called fetal acquisition. In many animals, fetal acquisition occurs as little animals nibble on their parents' feces. In humans, it's typically from nursing or from the birth process itself. When we talk about the microbes living together, we cultivate helpful microbes. People did this in the past by eating certain kinds of foods and all. We really do it now by taking cultures of certain microorganisms. But the whole goal is for the good microorganisms to take up space and take up food supply and to exclude the other microbes from growing at least too much. So two terms here: antagonism, the growth of good microorganisms works against the growth of harmful microorganisms. Competitive exclusion, competing for space and nutrients to reduce the population of harmful microbes.
You said competing for what, Mr.?
Space and nutrients. Food and a place to live. Or
Mr.
space
Moore,
to grow. Yes.
can you repeat the antagonism again, please?
Well, antagonism just means that you're trying to cultivate the good, helpful microorganisms so that they work against or exclude the harmful microorganisms. Antagonize, you work against them. Antagonism, competitive exclusion, it's two terms that mean similar things. Many organisms are opportunistic. So if we allow them to grow and to flourish, an organism that normally is not harmful can become so. Or an organism that would be okay in the gut would be harmful in the bladder or in the kidney. Certainly in the bloodstream, all organisms are harmful in the bloodstream. But so when we talk about opportunistic organisms, those that in normal circumstances are harmful, maybe even helpful, but under certain circumstances can become pathogenic. Don't write these numbers down, but would it surprise you that pathogenic organisms make up only about 3% of all of our microbes? Opportunistic organisms make up about 10% of our microbes. But that leaves 97% of organisms that in most cases are not harmful to us. I mean, that's the way we can live in a world that has tons and tons of microbes in it. It's that very few of them, percentage-wise, actually cause us disease. When we talk about drug resistance frequently comes from mutations. And we want to acknowledge that RNA viruses are more susceptible to mutations, and RNA viruses are the most common pathogenic viruses that we have. When we're talking about influenza type A, now notice that's very specific. If we talk about influenza type A infections, they mutate quite rapidly. It's called antigenic shift. If there are major changes in that H and N antigen, H and N are two antigens on influenza type A. A shift is a more dramatic change than a drift. So antigenic shift are major changes in the HN antigens. An antigenic drift are very minor changes in those HN antigens of influenza type A. When we look at drug resistance, many times microorganisms obtain factors from the environment or from other organisms. So a couple of terms here. Transformation means environmental uptake. In other words, one microbe dies, the other microbe laps up usually its plasmids. It could be its genes, but most of the time it's plasmids. And they will gain maybe drug resistance, maybe they'll gain some other kind of enzyme that makes them more pathogenic. So transformation is environmental uptake in bacteria. Transduction is what we've talked about over and over. That's the lysogenic infection of a virus to some other organism. It could be a bacterium, it could be a plant, it could be an animal, as long as it's not human. Because transduction is everything except humans. The same process in humans is called transfection. Another cause of drug resistance was improper use of our medications. I don't really going to ask you to go into what the improper use is, because it's really sort of implied in number 4. If we do the correct steps, We can perhaps reduce drug resistance. And the 4 steps have to do with finish your prescriptions, use drug combinations, limit the use of our medications, and certainly look for new drugs to add to the system. Your microbe resistance chart, I would definitely know the endospores are at the top of the chart. Prions are not very common. Endospores are. So spores are hard to kill. Viruses are fairly easy to kill. Gram negatives are harder to kill than gram positives because they have that outer membrane. Don't know how to learn that whole chart. Just learn some patterns in it. Won't be much about it. I'm not sure if it even made it to the party. I'm through. Your time.
Okay, I have a couple clarification ones. Starting off with number 9 for mRNA. Got the first copy of material is mRNA. Is that it? That one?
For prokaryotes. See what's in parentheses there.
Okay.
The first copy in prokaryotes. Now, the first copy in eukaryotes is not messenger RNA. So that's the difference.
That's the pre-mRNA, right?
Correct.
Okay. And then I had a couple more for number 30, the virulence factors, ability to produce what? Virulence?
Oh, a very ability to produce pathogenicity. I don't know another term. I hate the word pathogenicity because it's so.
Yeah, I must have just discarded that one. And then we need to know examples of each of that. Can we run through those real quick? Like an
I'm not going to ask you examples of each one.
Okay.
You can, I mean, they're good to know. Like flagella 0157, you know, E. coli.
Mm-hmm.
Fimbria, well, Fimbria is just attachment. There's several microbes that can attach. Capsules, it's self-explanatory. TB, tuberculosis has a capsule that's really hard to get drugs inside of. Enzymes, we mentioned things like lactamase and collagenase. Toxins, there's 2 types, exotoxins, endotoxins. We handle those both in different places. And phage infections, that's just lysogeny. That's about what you need to know.
All right, some more. I missed one thing on concept A, protein synthesis. I don't have anything for 3. So I have it happens, transcription happens at the nucleus. It's the template DNA. RNA polymerase binds at the promoter. What was the third one?
This is A on the left.
Yes.
RNA polymerase creates complementary copy.
Okay.
And then you add a 5' cap and poly A tail.
Mr. Moore, would you mind just reading the whole transcription part?
That particular one?
Yes.
Okay, look at it on the screen while we're talking about it. So you have template DNA, right? The RNA polymerase binds, and we know it has to bind at the promoter. The RNA polymerase creates a complementary copy. So there's your complementary copy. In finish, it adds you can't see the head, but it adds a prime cap and a poly A tail to complete messenger RNA.
All right, and could you repeat the translation as well?
Okay, so first, the messenger RNA right here is bound by the ribosome. Okay, so it snaps around it. The transfer RNAs bind their amino acids. See there? The messenger RNA codons select the transfer RNA anticodons. And of course, the transfer RNA has no acid with it. And the last step, what I said was, C messenger RNA code becomes an amino acid sequence. And an amino acid sequence, you could call it a peptide, or even call it a protein if you wanted to.
What was the one directly before that? Before the amino acid code becomes a sequence?
Messenger RNA selects the correct transfer RNA anticodon.
Okay, I thought there was one after that one. I thought there was one in between there. It was
So I made the statement that the transfer RNA has its specific amino acid with it.
That's more. So is there anything like in specific that you want us to know about concept E? I mean, yeah, with the E, like,
No, I mean, the only thing that I want you to realize about that is that the gene is not turned on if the product that it's going to work on is not there. That's what the whole part of the page from E. coli lac operon down, that's what that's about.
I apologize. I know that Jordan had already asked this, but on concept H, antagonism and competitive exclusion, can you repeat what you had stated on competitive exclusion? I have competing for space and I didn't catch the rest of that.
In competitive exclusion, you're competing for space and for food nutrients. That's the exclusion. See, if I can take up your space, you can't live there. Or if I can take up your food, you can't survive there.
Okay, thank you.
So for the one you just did about transcription, was that the concept or was that number 2?
It was concept, I think.
Okay, because number 2 or I'm sorry, not number 2. Yeah, under gene expression, they're kind of similar. So I just want to make sure.
Yeah, under gene expression. When you're talking about gene expressions, you're talking about transcription and translation. And in this concept, we were just picking out transcription and showing the parts of it, and translation and showing the parts of it. Yeah, they'll be the same.
Okay, so was there anything else for the concept A? Because I know there was 4 parts.
What I have is, for my 4 parts, I have, for transcription, you have the template DNA. You've got to identify, which DNA you're going to use. And then you have the RNA polymerase binds at the promoter. Third, you have the RNA polymerase creates a complementary copy. And then you create the 5' gap in the poly A tail.
Okay, was there something about transfer RNA in there? My notes all jumbled up on this one.
Transfer RNA has to be over there in translation.
Okay, I got that as the second. The transfer RNA binds the amino acids, right?
Yeah, see, transfer RNA is not in transcription.
Okay.
Look here at this sequence for a moment. Here is transcription up here at the nucleus. So here's your DNA template. Here's your RNA polymerase binding at the promoter. Here, creation of messenger RNA, and then here's the addition of your vectors. Now, this goes to translation, is down here at the ribosome. So here, your translation, your ribosome grabs around your messenger RNA, and the messenger RNA code pulls the right transfer RNA in, and there you're making your sequence. So just keep separate the components in transcription and translation, because they happen at the different place and they use different items to make it work. And that's what we've done on the left side and the right side of concept A. Just isolated the components together.
Professor Moore. I wanted just to make sure, just to clarify the information I have right for concept on the drug resistance. So for mutations, you have that the RNA viruses mutate more rapidly, is the most common pathogens, and antigenic shift is the influence of major changes in the HN antigens.
Yes.
And the antigenic drift is same thing, only in influenza, and makes minor changes in the HN antigens. Is it anything I'm missing on that part?
No, that was in the other test. I was going to show you a picture of a virus and show you what H and N stood for. I have it here. See these dudes right here? Some of them are H and some of them are N. And so what we have here, these are the antigens of a virus. And so when you're talking about antigenic shift, these things will change a lot. And they'll be different, they'll give them different identification numbers. But it's just a drift. They don't change very much. They don't change their names necessarily, but they just change the structure a little bit. And so it would mean that they would bind to a different animal, perhaps easier. So antigenic drift is not nearly as bad as an antigenic shift as far as changes go.
Okay, thank you.
You might repeat the exact line you said, because I heard part of that was incorrect from what I had down. It didn't have the HN on antigenic drift.
H and N are just the antigens. When you're talking about antigenic, you're talking about these receptor proteins out here. And major changes, like changing the numbers, is shift. And just minor changes in these would be drift. Where they not changed enough to actually change the numbers, they're just a little bit different.
Yeah, I heard her say the HN for when she read off antigenic drift. So I just wanted to say something about that. So she had that adjusted.
I got one more, two more. They, I don't know how to say it. The co-postulates.
Coach postulates, yeah.
I got same pathogen in every case of disease, diseased organisms, they isolate organisms, so on and so forth.
Yeah. See, the point of coach postulates is to make sure that you connect every case of a disease to the same pathogen. See, that introductory sentence is sort of the reason why you use it. Now, the postulates themselves is you have a case of a certain kind of disease. Whatever is infected, whether cow or person, you've got to isolate the infected disease agent, the pathogen, and grow it on a culture plate. Then you take that culture plate, take that microbe, infect a second host. Okay, when that host gets sick, you've got to isolate that same microorganism and grow it on a culture plate. So if all of those microbes match now, the one you started with and the one you finished with, then you have proven that that particular pathogen causes that specific disease.
You said once they what?
So I don't know if they
Just
the
that last
match.
sentence.
Say that again.
The last sentence you said if they match.
Oh, if the two microbes or the two pathogens match, or they are the same pathogen, then you have proven that that pathogen causes that disease.
Okay, thank you. I got two small ones. BRCA gene, did we have anything for that?
That's the breast gene. It's the tumor suppressor breast cancer gene.
Okay, and then the P55 or P53 gene?
P53 is the tumor suppressor gene that we all have a copy of, a single copy.
Okay. And then I'm missing the name for when the P53 is fouled up.
It's just a mutated P53.
Okay.
There's not a name for it.
Thank you. I didn't want to pick through all those videos to find all these random.
Oh, no, that's too much because those are small points. P53 is the one that the elephants have like 25 copies and they don't ever have cancer.
Oh yeah, that's right. I remember you made that comment.
I was actually teaching this particular component when that came out. It actually came out on Facebook about 2 years ago, 3 years ago. And I looked up, and it does, the research does support the fact that elephants have multiple copies of the P53. I don't know when they discovered P53 in humans, but it's been a while ago. What they find, people that have cancer, there's a high correlation between having cancer and having a mutated P53. And the same thing is true for breast cancer. There's a high correlation between having mutated BRCA genes and having breast cancer. It's not 1-to-1, of course, but it's just the risk, the incidence is higher if you have a mutated gene. But if I'm not mistaken, wasn't it Angelina Jolie that had double mastectomy because she had the quote breast cancer gene, and what she had was mutated BRCA genes?
It was her and Christina Applegate.
Oh, there was another one?
Yeah, they're the first two that fully came out stating that they had the mutated gene and then went and had their breasts taken.
I'm gonna have to ask if you would go over the spliceosome procedure. How do I exactly say that?
Well, yeah, spliceosome. I didn't really give you much procedure there, other than what you're saying. Let me put the picture here.
I had like large complex RNA proteins,
Yeah, I mean, spliceosome is a great big old bunch of proteins and enzymes. It's got RNA in it. It's just a huge complex. And I'm not caring that you know so much about the spliceosome, but what the spliceosome does is, like here you've got an intron with 2 exons. The spliceosome is going to take intron out and hook up the 2 introns together. So it splices them. Its name tells you what it does. It splices the introns back together.
Okay. I didn't know if that's going to be like a particular question for on concepts that was going to be longer.
I don't know. I can ask you what the spliceosome does to make it long enough, but not ask you what the spliceosome is.
Okay.
Yeah, that's what you said earlier. The question was, what does a spliceosome do?
Okay.
And I'm more concerned with the function rather than what it is. I don't know everything about what it is. You can look it up. I mean, but it's whenever you look it up, you don't know anything.
Okay.
I mean, it's just got parts to it.
Right. Okay. Thank you for that one.
It's just like, this is the picture that we draw about what a transfer RNA is. It has a place for the amino acid here, and it has the anticodon here. But what they really look like is something more like this. See, they're a sequence of messenger RNA. Not messenger RNA, a sequence of RNA. And here is the anticodon right here. So what something really is like is not necessarily what people draw it to look like. and their own little cartoon, you know, copy of it. It's just that that's probably too hard to draw in your books, so they make something simpler to be the symbol for it. But when you look at them under the electron microscope, when you look at a RNA, they actually have this kind of shape to them. And I don't think I have an actual electron micrograph of this. See, it's just a sequence of RNA.
Okay, I appreciate you going back over that.
What else?
Not too much else on that. The other things what I've waited for really. Because you probably don't mind if I end up redoing the chemical part on the last summary. And then I was just double checking information about testings and stuff, but you know, I wait for that information. Just due to certain circumstances that I knew I'd have to talk to you about later.
Okay.
Can anyone who recorded tonight send me the video, please?
This is Trish. If you want to text me your email, I'll go ahead and send you a copy. I usually just post it to like my YouTube thing and send you a link. My phone number is 512-481-8847.
Thank you.
I'm going to send your phone number to that car warranty thing that keeps calling me. So they'll call you instead of me.
That's not even nice, Professor.
Please don't do that because my previous job still has me listed as the emergency contact for when people have a toothache or whatever. And so it's still coming to my phone after hours and I don't work there anymore.
No.
Good night, that's more.
Good night, Chris.
Have a good night, Professor.
Good night. See y'all.